Hi, I'm Paul Pryor, Executive Director of University of Arizona's Yuma Center of Excellence for Desert Agriculture. We're a public-private partnership proactively funded by members of the desert agriculture production industry to solve the problems that they face. Speaking of pressing problems, Fusarium wilt of lettuce is a soil-borne disease that is causing more and more destruction of valuable lettuce crops every year in the Yuma area and increasingly in California production areas. The Yuma Center of Excellence is working with several researchers to attack this problem on many fronts, including cultivar selection, evaluating and advancing crop protection products, enhancing soil health, and improving diagnostic techniques. Some projects hope for results that are useful to researchers searching for new solutions, while others aim to give usable results to producers as they try to mitigate Fusarium's impact on yield and quality. With specialty crop block grant funding from the USDA and Arizona Department of Agriculture, we partnered with Dr. Barry Pryor, a research professor and plant pathologist in the University of Arizona's School of Plant Sciences, to explore new techniques to improve greenhouse research and develop DNA-based methods for rapid detection of the disease, in the lab, in the greenhouse, and even in the field. Dr. Pryor performed his research in greenhouses and laboratories on the Tucson campus of the University of Arizona. I'm excited to have him share his results with you next. After you finish watching this video, I hope that you'll take time to watch the accompanying instructional videos that detail the methods for extracting DNA and processing it for rapid diagnostics. We also have information and videos from our other research projects on our website at desertagsolutions.org. So without further ado, let me hand it over to Dr. Barry Pryor to give an overview of his research and results. Barry?
Thank you, Paul. At the University of Arizona School of Plant Sciences, we are conducting research on the management and control of Fusarium wilt of lettuce. Increasingly, we're utilizing the greenhouse as a platform for growing lettuce and conducting research similar to the kinds of studies we might perform outside in the field. Now, although the greenhouse will never replace the field for certain types of studies or certain kinds of experiments, it does offer a number of advantages for conducting research on diseases of lettuce and other vegetable crops. First, in the greenhouse, we can control the environmental parameters on multiple levels. This is a huge advantage over field-based studies in which we need to accept the kinds of weather that Mother Nature delivers. We can control the temperature and the humidity, the light period and the intensity, of course using supplemental lighting. We can more precisely control the irrigation quantity, quality, and frequency, and more precisely control nutrient delivery. Soil characteristics can be varied from completely artificial soil to importing actual native soils from the field. And we can do all of these studies free from insects or other crop pests. Now second, in terms of research on Fusarium wilt management, we can use native strains of Fusarium present in imported field soils and using the population densities naturally present. Or rather than be limited to natural infestations, we can use clean soil and introduce the pathogen to this soil at very precise densities. Moreover, we can use different strains of the fungus or even combinations of the strains of the fungus to create disease symptoms and disease development more precisely needed for our experiments. Here are some examples of lettuce inoculated with different strains of Fusarium exhibiting a range of symptoms from wilting, chlorosis, stunting, and severe necrosis. Third, and very importantly, in the greenhouse we can grow lettuce nearly year-round. Whereas in the field, we might be able to get 1 or 2 experiments conducted per year, in the greenhouse we can grow up to 6 to 8 lettuce crops, depending on the level of maturity we need to bring the lettuce plants to. As such, we can conduct our research year-round, and this includes experiments on fungus strains virulence, lettuce cultivar tolerance, and testing materials for disease suppression. Now ultimately, all of these findings will need to be ground truth out in the field for final evaluation. But by conducting many studies in quick succession in the greenhouse, we can move quickly and zero in on the most promising management strategies that can then be tested in the field for durability and optimal utility. Much of our recent research is focused on early and precise detection of the Fusarium fungus in plant tissue and in soil. We are looking at advanced methods of detecting the pathogen DNA at very low concentrations, with the goal of detecting infestations as early as possible. Moreover, we are looking at methods of detecting pathogen DNA that might be able to be used in the field, rather than bring the samples back into the laboratory. This would enable the grower to make in-field diagnosis of fungal infection, which may enable decisions on crop management several days earlier. We've produced a number of instructional videos to present some of these advanced Fusarium detection techniques that we've been developing and evaluating.
The first video shows how to extract the crude DNA from suspect lettuce plants and how to use the AgDia Amplify RP system, a very simple and quick method of detection that can be used directly in the field. The second video shows how to further purify that crude sample, which can then be used for other types of analyses in the lab. And the third video shows a related detection method called LAMP PCR that can be initiated in the field with sample collection and DNA processing, but then having samples returned to the lab for further sample purification and analysis.
Both methods are highly sensitive and work well under most conditions. And we're continually optimizing these methods to improve their sensitivity and ultimately their utility in the field or in the lab. Please enjoy these videos demonstrating an introduction and step-by-step instructions for using these novel diagnostic methods for detecting Fusarium wilt of lettuce.